Welcome to episode 8 of the Ada Lovelace Day podcast. My name is Sue Charman-Anderson, and every month I talk to women in STEM about their work, as well as talking to women and men about historic and modern women's achievements, discoveries and inventions. Our podcast is made possible thanks to the generous support of ARM, our exclusive semiconductor industry sponsor. You can find out more about ARM's energy-efficient microprocessor technology on their website at arm.com, and you can follow them on Twitter, at ARM Holdings. If you'd like to join ARM as a co-sponsor of our podcast, then please email me at suw@findingada.com. This month, we're talking to Dr. Rae Robertson-Anderson about biological soft matter and biomaterials. And we'll be chatting with Ecologist Dr Thorunn Helgason about Barbara Mosse and how women's contributions to STEM are often overlooked. So this month I'm talking to Dr. Rae Robertson-Anderson, Associate Professor and Chair in the Department of Physics and Biophysics at the University of San Diego. Hi, Rae.
Hi.
So, Rae, physics and biophysics, tell me a little bit, which side of that do you fall on?
Yeah, I actually fall kind of in between them. So biophysics, you're applying physics concepts and techniques and problem solving to questions in biology. You know, why do certain molecules bind to other molecules? How does DNA, how do viruses infect cells, and these types of questions. And I'm very interested in using these molecules to understand and design new materials. So I'm really interested in the molecular properties and how, when you have a whole bunch of these biological molecules all together, how they create these really new, fascinating materials. Hardcore biophysicist wouldn't call me a biophysicist, and a hardcore physicist might not call me a physicist, so kind of in between.
So you're really sort of on that intersection between physics and biology.
Right. So there's this whole field of, soft materials and biomaterials where a number of us feel kind of in between these two fields where we're, particularly I'm looking at large, complicated biological molecules like DNA and the proteins in our cells, or in our cytoskeleton, so actin and microtubules, and these really long, complicated molecules, when you have them at really high concentrations, they create these really fascinating materials. But, a biologist would say, well, who cares? If you're not recreating what's inside necessarily the cell or learning anything about the cell, then maybe it's not important. And the physicist would say, oh, you're using biological molecules, so it's not hardcore physics. But I think it's great fun. So all the work we do is using molecular level techniques to study these things. So we're using fluorescence microscopy and optical tweezers to actually look at single molecules in these complicated environments and materials and see how they move and see how they interact with each other and measure forces at the molecular level. So still, every day, being able to just see a DNA molecule or a long actin filament moving around and actually measuring a force when you push on it, how it's responding, that to me just still is fascinating.
So, of course, when you say soft matter and biomaterials, there's a part of my brain that kind of goes, you mean sort of skin or kidneys or muscle, and that's not really exactly what you're talking about, is it?
Well, soft materials right now in industry, like in athletics and in the military, people are trying to design these materials that have multiple properties that can behave different ways depending on how they're strained. So something that's really high strength but also very lightweight, or a material that can behave partially fluid-like and partially solid-like. So a perfect example when I'm describing soft materials is oobleck, or if you've ever mixed cornstarch and water, which some people call this oobleck. But cornstarch is like these long biological molecules that I study, like DNA and actin and microtubules. And so they're these large polymers, and when you add water to them, then it becomes this viscoelastic material that if you just try stirring it slowly, it seems just like a fluid. But then if you try to, say, punch it or throw the stuff really hard, it tenses up like a solid. lot of materials in biology behave like that or have those similar properties, that it's very hard for us to actually recreate those with just synthetic materials. So there's a big push to understand how biology can produce these materials so effectively, and how we can understand why it is these materials behave this way and how we can reproduce them. So, I mean, when I talk about soft materials, yes, I mean, you can think, so everything in our body, cells and muscle and kidneys and all of these things are like soft, gushy materials because in some sense they can flow like a fluid. Even your blood has this viscoelastic property that these things can kind of flow. If you try to push on muscle or organ or something like that, it'll deform, but it also can behave solid-like. And so understanding this sort of interplay between solid-like behavior and fluid-like behavior, and sort of how they behave differently depending on how you're straining them is what I find really interesting. And not just saying, okay, macroscopically, what I'm more interested in is not just that question, because then you could just study a whole bunch of materials and basically tabulate how all these things respond to certain strains, and that's great. But if you want to actually design materials based on what you find, the question you want to ask is what the molecules are that make up these materials. What are they doing? How do they interact with each other? What forces do they impart on each other? How are they stretched or strained? And how do they relax? And how is that related to the molecules themselves? How does that vary depending on, the size of the DNA that's making up your cells, or the size of, how much, the concentration of the actin filaments you have in your cytoskeleton? All these questions. So I'm much more interested in what's happening at a molecular level, so that then we can build up. So it's a very kind of bottom-up approach.
And so then from that understanding, you're creating these biomaterials. Are these sort of completely new materials, or is this sort of replicating what nature creates?
It's a little of both. So, like, for instance, the cytoskeleton in our cells it's made up of actin filaments and microtubules and intermediate filaments. And for instance, all of these, actin and microtubules range from maybe 10 microns in length, up to maybe 40 or 50 or even longer. And microtubules are really stiff, so they have a persistence length of about 1 millimeter. And actin is still stiff, but it's the persistence length is on the order of microns. And so that means together they provide different rigidity and different abilities in the cell. And also these molecules can polymerize and depolymerize, so they're all monomers. And the way you get these long filaments that maybe are 10 microns long, which in the biological world, that's big. And if you're talking about a biological molecule, a single molecule that, has a persistence length of 1 millimeter, that's huge. But so because of in our cells, so we know the cytoskeleton in our cells has these really interesting multifunctional properties where it can, at some sense, it can be mobile and fluid-like. In other senses, it can tense up. And that can change based on the concentrations and the polymerization states of these cytoskeletons. So we use that knowing that when you have this combination, you're going to get these interesting properties. We make materials, fluids that are comprised of different ratios of actin and microtubules. And then we use optical tweezers to measure the forces at the molecular level. So, varying the ratios of these things, then saying how are they responding more elastically or more fluid-like, depending on how you're pushing on them, and then what are the different relaxation mechanisms? Does that depend on the length scale and the time scale that you're applying this perturbation? And so we're using insights from biology to create new materials. But then there are other materials that we're interested in that don't exist really in biology. Like if you just have DNA. So DNA is it can also be very long. So we study DNA molecules that can be up to 40 microns long, but their persistence length's on the order of a nanometer. But now when you have these things in really high concentrations, they also create really cool multifunctional materials. And so you can think about, when I think of these DNA systems, think of like a pile of spaghetti. So they're really these long, skinny, flexible molecules that are all really highly entangled with each other. And so you pull on one, and it's going to affect all the other surrounding strands of spaghetti. And so, how does that respond? If you pull on one, how many strands of spaghetti are actually affected when you just tug on one? And so that's some of the things we can actually ask, using the techniques that we use. So yes, I think there are systems that are mimics of what's in biology, and also systems that are just using biological molecules to create completely new materials that you don't find in biology.
So these materials, you're talking a lot about optical tweezers and molecular-sized stuff. That's a technical term. Does this scale up? Does this wind up eventually as a material that I can pick up and hold?
Yeah, absolutely. And that is the goal. And all these materials at this stage can be scaled up. But first, we need to understand how, what happens when you scale them up. And so we're looking at studying these things at the molecular level to understand when you vary different molecular properties, how that affects the material at a small scale. But then we can also, once we understand that at the molecular level, we can scale it out.
And what's the end game? What are the real world sort of applications of these new materials? Some
of the funding I get for this research is from the Air Force. And so some of the things the Air Force is interested in is learning from biology to create these new materials. And, so for instance, one application is, if you imagine, like right now, bulletproof vests are like quite heavy. And if you have some material like oobleck, say, this cornstarch and water, or these materials in biology that are very lightweight and almost like fluid-like, but then if you just kind of push on it, it would flow. But then if a bullet was shot straight at this material, that strain rate is so high that it would cause it to tense up. You can apply this type of thinking also to just other military, like tanks and airplanes and things like that, where they have to have really high strength but very lightweight. And the same can be thought of. So the athletics industry is also really interested in these types of things. Everyone's trying to design the best shoes and the best helmets. Everyone's interested in designing better helmets that can be much more resistant to injury, but also maintain some comfort.
When it comes to your day-to-day work, how does your day normally sort of pan out? Because it's I can kind of get a fairly easy grip on sort of how an engineer works and the kinds of stuff that they do, but how does your day work?
That, I guess, depends a little bit on the time of year. I'm a professor here at University of San Diego, and it's an undergraduate institution, predominantly undergraduate. So most of the students doing research in my lab are undergraduates. I also have a few postdocs. And during the semester, we were teaching. And at mostly undergraduate institutions, you teach more than at R1 or big research universities. So, and plus as chair, I have a lot of administrative duties, meeting with staff and other things like that. So my days, this is actually what I really love about what I do, is because I get to do all of these different things. There some days during the summer when I don't teach, and then I'm just in the lab a lot. And what I really love working with undergraduates. That's why I actually came to an undergraduate institution. Not so that I could teach more. I really enjoy teaching, but so that I could do research with undergraduates. Because it was my undergraduate research experience that really made me realize that this is what I wanted to do for a career. So during the summer, I'm in the lab as much as possible, working with the students and with my postdocs, and also working on, writing grants and writing papers. And during the semester, it's much more hectic. My mind. So during the summer, I like to think of it's like when I was a postdoc, where I could actually just kind of think about a couple different things. During the semester, it's much more crazy because I usually teach 1 to 2 classes a semester, and then I have office hours, and I still have students doing research during the semester. So yeah, it keeps it exciting because things are always, every day is different, and there's always, with undergraduates, they're not there as long as grad students, and they're not there all the time. So every day, I might have one student in and not the other, or I might teach one class, and then the next day I might, have a whole bunch of meetings or something. But I mean, the meetings being chair, I really enjoy that too, because I actually get to think about how we can improve our department, and how we can improve education of our students, and the university as a whole. So yeah, just everything. I find my job very exciting, but I have to say what I love most is working with the undergraduates. Most of my papers I publish with the undergraduates, and when they're first author, they are taking the helm of writing the paper, not just doing the experiments. So it's really fun for me.
It does sound, I mean, the undergraduate research aspect, sounds absolutely fantastic. And I think an important way to get people to feel a degree of ownership about the science that they're doing. You're not just an empty vessel waiting to have information poured into you. You're actually engaged in creating new knowledge. And that sounds absolutely fantastic. I would love that.
For me, it seems like, especially now, in this day and age with Google and everything, it seems like just giving our students knowledge is not really what it used to be. And what I really think what we need to be giving our students is the skills and the confidence, and that sense of ownership, that that aha moment when they're in the lab and they figure something out, and they're actually understanding what's going on and developing these skills that they can take on to graduate school. I mean, that's something, or to their careers or anything. I mean, that confidence and the skills they build, that's something you can't get from Google. And I feel, for me, as an educator and as a scientist, that's my main purpose, is not to give these students knowledge, but I mean, that's important too, but to teach them how to think. Because as scientists, I mean, yeah, we all come from different, people in STEM, we're all studying different things, but I think what is similar between all of us is, at some point along the way, we learned how to think critically, and how to solve problems, and it's those things that we still really love. It doesn't matter whether you're studying black holes or studying DNA. We have the same mindset and approach, and that's what we really want to get our students.
So how did you end up in biomaterials? What was your path to where you are now?
When I was an undergraduate, I did research, and I studied granular material, which people don't really study much anymore, but I was basically studying diffusion of little vibrating steel spheres vibrating on this plate, and how the diffusion varied depending on vibration. But learning all about diffusion and velocity autocorrelations of these complicated systems made me realize how much I really enjoyed statistical mechanics and looking at transport properties, and I also learned a little bit about coding and things. So I thought, I really liked that type of physics. And I guess starting before even talking about my research, I guess it was in high school where I really just determined that I loved physics. I really enjoyed science, I really enjoyed math, and then when I took my first physics class, I felt like, oh my God, this is a dream come true. This is science and math combined into one thing. And I just thought it was the coolest thing ever, and I realized, I was fairly good at it. And so then when I went on to college, I still thought when I went to college that maybe I would find an interest in something else, but I still took physics classes, and the more I learned, I just loved it. And then it was really my chance to do research, where I really realized this is what I wanted to do. And then one of my professors told me, why don't you go to grad school? And I said, well, but I think I should probably go get a job. And he said, they pay you to go to grad school. And this blew my mind. I thought, like, wow, really? I mean, I could keep learning physics and keep doing research, and somebody would pay me to do it? And I don't know, that to me, I was just sold, because I was not ready after 4 years in college to stop learning about physics. And I realized I really enjoyed looking at more complicated statistical mechanics problems with, many, many particles, and how do you sort out what's going on. And so a lot of people back then who were studying granular materials started looking more at polymer physics and seeing polymers, these long stringy molecules, just like what I'm studying now, the DNA and actin and microtubules and all synthetic polymers. And when I went to graduate school at UC San Diego, I met with a faculty member who became my PI, and he was studying this polymer physics and looking at diffusion coefficients and transport properties. And then he showed me how he could study these things by looking at the molecules themselves. And this, like when I actually saw a DNA molecule, labeled, and I could see it wiggling around, I thought, like, this is amazing. It kind of combined both of my interests, my interest in statistical mechanics questions, but also this fascination with being able to actually see and almost touch or just like push or pull on these individual molecules. I thought it was just fascinating because it seemed you have this very, very complicated system and then say, pare it all down to what is the least complicated way we can study this. Well, let's just look at a single molecule in there and what's it doing. And as physicists, we try to always simplify things, and so to me this was just a really beautiful way to simplify complex systems. So through my PhD research is where I really developed my enjoyment of actually using single molecule approaches to study these big complex systems. It's still great fun. I still have more and more projects that I want to do and I wish I had time to do. More materials I want to study, more molecules I'm interested in, and more collaborations that I want to get involved in. And it's just, if only there were more hours in a day.
I think we all feel that particular pressure. It's wonderful to hear how passionate you are about what you do, and it seems to me that your excitement for what you do hasn't waned in the slightest, just gotten more and more, which is always lovely, because I think, sometimes I think, the idea of having a career gets a little bit of a bad rap and people think they're going to get bored, but there's always something exciting on the horizon.
Yeah, it's so true. I mean, yeah, like I said, if there were just more hours in a day, because I have so many projects in my head and so many things that I want to do, and every day it's so much fun. I just love coming and walking into the lab or walking into the classroom, and yeah, I can't imagine getting bored with it.
Well, thank you so much for talking to us, Rae. It's been really, really interesting.
Yeah, it's been quite fun.
Excellent. And if you want to find out more about Rae and her research, there's a link to her research website in our show notes. So today I'd like to welcome Dr Thorunn Helgason, a senior lecturer at the University of York, to the Ada Lovelace Day podcast. Hi, Thorunn.
Hi, Sue.
So, Thorunn, you're an ecologist, but ecology is a pretty broad subject. Can you tell me a little bit about your work?
Okay, so ecology is a very broad subject. It's basically the study of organisms and where they live and why they live where they do. That's fundamentally what ecology is. The group of organisms I work on are a particular group of fungi called the arbuscular mycorrhizal fungi. They're not mushrooms and toadstools like we're familiar with in pictures and that you see in the woods when you go out and looking for them. They're a very boring looking bunch of fungi that only grow as filaments in the soil, and they also grow inside plant roots.
Right, and why are they important?
They're really important because they're what's called a symbiosis. They're actually exchanging nutrients between plants and the soil. They're a kind of keystone link between what goes on in the soil and what goes on in the plant.
Now, normally at this point in the podcast, I would be asking you to name the woman in STEM whose work we are about to discuss. But this conversation is not going to be quite that straightforward, is it?
No. So when you asked me to do this podcast, it struck me that I have very few, if any, female role models that I've experienced in my career as a scientist. There isn't one person that I can point to and say, I was really inspired by that person that woman to do what I'm doing now. I did my degree in an agriculture and forestry department, which is a very male-dominated subject, certainly was at that time. And I was on a science campus where most of the students were male, most of the staff were male too. And while I was doing my bachelor's degree, I got vanishingly few lectures at all from other women scientists. So I didn't have obvious role models to choose from.
But of course, as many scientists are fond of saying, absence of evidence isn't evidence of absence. There were and there are women in ecology making important discoveries.
Absolutely, absolutely. And what happens when you work in a discipline, and the work on mycorrhizas that I've been doing, is you read a lot of papers, you go to conferences, you see the women who are working in the field and they're presenting data, and there are some really important women in the field of mycorrhizal research who've been working with these fungi for a long, long time. I mean, the history of the research on this particular group of fungi is long. They were understood and known about. People could see them under the microscope in the early 20th century, but they couldn't identify who they were, they couldn't put a name to them, and they didn't know what they were doing. And absolutely fundamental to our understanding of these organisms now was the work of a woman called Barbara Mosse. And she worked for many years as a microbiologist at the Rothamsted Research Station in the UK and at East Malling Research Station, also in the UK. And her work was absolutely fundamental to the science as we know it today.
And now you mentioned Barbara Mosse to me yesterday, and I went to look her up on Google, and it was really difficult to find any information on her at all.
That's right. There's as far as I know, there's no Wikipedia page on her. There's no biographies of her that I could find. She's mentioned there are a number of papers that talk about the history of mycorrhizal research, and her work is mentioned there. I mean, what's really interesting is that the foundational thing that she did was to prove that the reproductive structures of the fungus, the spores, that's the sort of equivalent of seeds in fungi. They don't make seeds as we know it from flowers. They make these things called spores. And she proved that the spores that people were finding in the soil and the structures that people could observe in the roots, she proved that the spores caused those structures. So she was able to isolate those spores. She actually grew strawberry plants with these spores, and having grown them in pots for a while, she was able to show that indeed the structures that we knew about from microscopy had been caused by these fungal reproductive structures. And as a result of that, people were able to start to put names to them. And the fungus that she worked on actually bears her name. It's had a number of genus names. It started off as Endogone, became Glomus, and is now Funneliformis, but it carries the species name Mosse after her work. So it was quite a while before I realised that Mosse actually referred to a woman scientist.
And it's understandable to some extent that someone like me, I mean, fungi is I know very little about fungi apart from which ones I like eating, that I might not necessarily have heard of Barbara Mosse, but it seems a little peculiar in a way that it took you so long to stumble on her, and you had to stumble on her. She's not someone that you could find even if you're looking for her.
I think that's absolutely true, and it's the invisibility of it. She's by no means the only woman who's worked, only woman of her generation who worked on these fungi. There are many of them out there, and many emeritus professors like Lynette Abbott in Australia. There's Sally Smith also in Australia. And, you know, there are many women out there who've been working in this field for a long time, and we don't have the directories and resources, and it's celebrating them and putting faces to names. Perhaps it's part of science that we tend, as scientists, to want to be valued for our work, and that we as scientists aren't aware of how important we are as role models to people coming up behind us. So, you know, perhaps we're a little bit shy to be ourselves, and therefore it's relatively easy to become a little bit invisible as a person. So maybe we should not be so shy about our work.
So I wonder if part of the problem is that with tools like Wikipedia, it very much depends on the editors deciding who gets a page, and where there are areas of science that have a clear public interest. A lot of the health work that's been done, for example, medicine, surgery, human biology, everyone's really interested in that. When it gets to sort of obscure fungi, is it just that there's not enough interest, that it's such a specialised area that people are like, it's not important?
Absolutely, you're absolutely right. In the research that I'm involved in at the moment, a lot of it's based on sustainable agriculture. The crosstalk between the arbuscular mycorrhizal fungi and crop plants is a really important part of maintaining crop yields in a changing climate and where soils are degrading and things like that. And we talk in our research a huge amount about public engagement, about getting people interested in the output of the research and how important agriculture is and climate change and all of these issues, and public engagement is a key part of research. Perhaps what we don't do so much is also introduce the people who do the work. And perhaps it's definitely part of Ada Lovelace as a movement to celebrate the people who do it. And I think perhaps in the public engagement agenda in science, we talk so much about impact and telling the world about the impact that we're having. Perhaps we also should spend some time saying, and these are the people who carry it out. These are people like you. You could do this too.
It makes the science more approachable when you are coming at it as well from a point of view looking at the kinds of people who've done the work. And it gives you just a different angle to look at it, because when you think there's another woman in mycology whose work has been sort of pretty much glossed over, but she's a household name for completely different reasons.
So I think you're talking about Beatrix Potter, aren't you?
I am indeed.
Yes. Beatrix Potter, we all know from her canon of children's literature and her excellent drawings, but she was a brilliant artist, as we all know, but she was also a brilliant natural historian, and she had a particular interest in fungi. And she collected many of them, she drew many of them, and her drawings of fungi, which are held in the Armit Museum and Library, are absolutely stunning. And yet it's not something she's well known for at all.
And I think there is an increasing understanding of Potter's work, and it's rather like people like Florence Nightingale, who's known for nursing rather than being a statistician, that it seems that we kind of erase women's contributions to science because they don't fit into the stereotype of what a woman should do.
I think that's true. She tried very hard, Beatrix Potter tried very hard to get involved with sort of the formal botany community. She was speaking to people at Kew Gardens and at the Linnaean Society of London, and these were the institutions that were the gatekeepers of 19th century botany in Britain at the time. But these were male-dominated environments, and they didn't accept her work.
Yeah, and she wasn't the only woman to be excluded from those kinds of societies. But that exclusion has even in more modern times, with people like Barbara Mosse, whilst they might have had entrance into scientific societies, there's still this cultural pressure to downplay their work.
I have to be honest and say, personally, I haven't experienced that. I think within groups of scientists, as I said earlier, we're predisposed to respect the work, and, the work itself often stands up to scrutiny. It's whether the person who did it is recognised for it, I think. There are other examples of women, because women were not allowed for a long time in the 20th century to actually graduate with bachelor's degrees from any of the old institutions. As a result, their work was downplayed because they were the technicians on the project, and yet they did most of it and wrote most of it, but they couldn't publish it under their own name because they couldn't get the degrees. I do confess to be thinking of a particular woman who was instrumental in plant population genetics, but I confess I can't remember her name right now, which is exactly part of the problem, isn't it? I know she's there, but I don't know who she is.
It is. It's a massive part of the problem. So I think we can't rely on our collective, our group memory for understanding the true extent of the contributions of women in STEM. And I think, one of the things that you often see is that where women are not represented, they're assumed to not exist. And I think Mosse is a great example of how, that's just simply not true. So I kind of feel like we need to put a lot more effort into digging into the history of even sort of relatively compact fields like this sort of fungal ecology, and find the women whose work has been, the work is respected, but they have been sort of erased from the record, as it were.
They may not even have been erased from the record. They were simply never put on the record. And I think that would be another way to look at it. But I think that's right. I think if we are truly to celebrate women in science, then we as scientists, we in the scientific community, should look into the past of our discipline and see who they were and find them and celebrate them, so that the disciplines look less male-dominated and a truer reflection of the people who actually did the work.
Absolutely. And I think one of the challenges with this podcast, and particularly this segment, is you say to people, I want you to talk about a woman in STEM. And like you, a lot of people go, I'm not sure. And I think the lesson here really is like, if you can't name a woman in your field right now off the tip of your tongue, you should go and do a little bit of digging, because there's almost certainly a woman out there who's not getting the recognition she deserves.
I completely agree, Sue. I completely agree.
Well, thank you so much for talking to us, Thorunn. It's been interesting to learn about Barbara Mosse and Beatrix Potter, but also I think there's an important lesson to take away from this.
I agree. I think we should all go and look at our papers and look into the names of the papers that we like the best and find out about the people who actually wrote them.
Fantastic. Thank you so much.
It's a pleasure, Sue.
And you can find out more about Thorunn Helgason on our website. We have her biography and notes to her publications in the show notes. Thanks to Rae and Thorunn for talking to us this month. If you want to find out more about them and their work, just visit our website at findingada.com/ep8/. Thanks again to our sponsor, ARM, for their ongoing support. If you'd like to join them as a podcast sponsor, please get in touch. Thanks also to our editor, Andrew Marks. Don't forget, you can subscribe to our podcast on iTunes, Google Play and Stitcher. But if you find yourself at a loose end over the holiday season, why not work your way through our archive? Our past guests have included bubble physicist Dr. Helen Chersky, spacecraft structures engineer Abby Hutty, technology journalist Clive Thompson, and astronomers Professor Hayley Gomez and Dr. Karen Masters, to name just a few. You can browse through all of our episodes at findingada.com/podcast. And if that's not enough, we've also got this year's Ada Lovelace Day live talks up online. You can hear from design engineer Yewanda Yakinola, planetary physicist Dr. Sheila Kanani, science writer Dr. Kat Arney, developer Jenny Duckett, mathematician Dr. Sarah Santos, computational biologist Dr. Bisan Al-Lazikani, and climate scientist Dr. Anna Jones. And if you still haven't had enough, we've also got 2 fantastic biographical anthologies featuring some truly amazing stories. A Passion for Science, Stories of Discovery and Invention, features astronomer Dame Jocelyn Bell Burnell, who discovered pulsars, nuclear physicist Chien-Shiung Wu, and geologist Dr. Florence Bascom. Our second book, More Passion for Science, Journeys into the Unknown, tells the story of the Air Transport Auxiliary, who moved planes from airbase to airbase during the Second World War. It also explores the work of inventor Martha Matilda Harper, physicist Joan Curran, and statistician and political campaigner Florence Nightingale. Both ebooks are available for just $1.99 from Amazon, and there are selected chapters available to read for free on our website. Just visit findingada.com/shop. Next month, we've got Professor Elaine Chu explaining how she uses mathematical and computational models to reveal the hidden structures of music. And we'll be talking to marine biologist Dr. Helen Scales about the pioneering work of the shark lady, Eugenie Clark. So join us in 2017 to find out even more about amazing women in STEM.